that's the shape of a microtubule. Whereas actin filaments are a little bit smaller, and they are not hollow tubules. They are like two braided chains, two intertwined chains of these smaller proteins all linked together. Any cytoskeleton questions? All right, let's see. Now, so there is one microtubule. Imagine that you had several of these microtubules, and you're going to arrange them into a cylinder shape like this. You take another microtubule and put it there, another one 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 there, like that. So here you have a larger structure, cylinder-shaped structure, made by fastening together several microtubules, and that structure is found inside cells. It's called a centriole, or sometimes a basal body. Here we go. A centriole or a basal body. Now, just to simplify my diagram here, instead of showing it like that, I'm just going to show it as kind of a solid cylinder like that. But just bear in mind, it's made out of a ring, a cylinder made out of several microtubules joined together. All right, so what these centrioles serve as are organizing centers or anchoring points for microtubules. Microtubules by themselves can be very long and stretch all the way across the cell, but those microtubules wouldn't be able to function very well as a solid support beam, so to speak, unless there was an anchoring point for them, and that's what the centrioles are. To make an analogy here, here's the Golden Gate Bridge, and if you think of all these cables and things that you see here as being like microtubules of the cell, well, these big concrete blocks serve as anchoring points for many of the cables of the bridge, and so think of these as the centrioles of the basal bodies, and again, the cables are the microtubules that extend outward from them. Here's an example. There's a sperm cell, and there's its flagella sticking off to this side right here, and as I said earlier, what gives the flagella its structure are microtubule proteins inside of it. Well, to keep those microtubules organized, to keep them from drifting apart from each other, at the base of every flagella, you'll find one centriole, like that, hold them in place. And the same is true with the cilia also. Each cilia has a centriole at its bottom to hold its microtubules in place. And it's not just at the bases of the flagella and the cilia. All microtubules that you see inside the cell are anchored by centrioles. When we discuss cell division, which is called mitosis, what we're going to see is a very important role for those microtubules and for the centrioles that hold them. The microtubules when the cell divides, reach out and latch on to this middle part of the chromosomes called the centromere, and then end up pulling the chromosomes towards opposite ends of the cell, opposite poles of the cell when the cell divides. Alrighty, well, the last of the non-membranous organelles are the ribosomes, and we already covered those guys. Remember that the ribosomes are the organelles that make proteins. They grab the amino acids and assemble the proteins out of the amino acids. The ribosomes get their information from the messenger RNA. Comes out of the nucleus. And then by reading the messenger RNA, they assemble the protein. The ribosomes themselves are made out of proteins and RNA. They're kind of a mixture of protein and RNA. All right, you guys may be done with organelles of the cell. So there's a lot going on inside a cell. You have membranous organelles, non-membranous organelles, including the cytoskeleton. It's a complex world in there. Any closing questions on the cell? Don't get up. We do have 7 more minutes. I want to go on to the next topic, but any cell questions?
What was it that you mentioned about ribosomes? Because I didn't really understand that part.
Oh, sure. The ribosomes, which you usually find clinging to the outside of the rough endoplasmic reticulum, their job is to make proteins. The amino acids are the building blocks of proteins, and the ribosomes grab the amino acids and link them together to make proteins. And the ribosomes are an example of a non-membranous organelle. They're not made out of phospholipid bilayer, they're made out of proteins and RNA, and you find them both in prokaryotic cells and eukaryotic cells. Everybody's got ribosomes. That'd be a good t-shirt. Everybody's got ribosomes. All right. We're going to start a new topic now called energy metabolism. And remember that this will not be on the midterm coming up next week. Let's see. When I was introducing proteins, I showed you this slide right here. And I said, if you think of the cell as like a factory, the proteins are the workers. Proteins carry out all the tasks that keep the cell going. They are the active, doing action molecules of the cell. They carry out all the work and all the tasks. And like this guy was supposed to be a transport protein for bring things through the cell membrane. This guy was a receptor protein. But these guys, I think, are the most important of all. Those are supposed to be the enzymes, and they carry out chemical reactions. They change molecules. And it takes a lot of chemical reactions to keep a cell alive. There are thousands of different organic molecules that float around inside cells, and the cell constantly needs to be changing some of these organic molecules into different ones, and then taking those molecules and changing them into different ones still to carry out all the life processes. And in a way, the definition of life is just a self-sustaining chemical reaction going on inside a cell. And remember, each enzyme is a specialist in working with only one particular molecule and carrying out only one particular chemical reaction on that molecule. So since the cell requires thousands of different chemical reactions to be constantly going on, the cell requires thousands of different enzymes floating around the cytoplasm, and that's what you see. There was a chart I saw somewhere that showed not all the thousands of chemical reactions, but just some of the chemical reactions going on inside cells, and I took a picture of some of that chart. These are some of the organic molecules inside the cell, and the little arrows represent the enzymes that change one into another. And I don't know what most of these molecules are. This one's L-xylulose, so there's some enzyme that takes that and changes it into L-xylulose 5-phosphate, and then there's another enzyme that takes that and changes it into L-ribulose 5-phosphate, and that, I guess, can be changed into ribulose, which can then be changed into diarabinitol, which can somehow turn into 3-oxygluconates, whatever that is. I mean, just look how complex it looks. And this was just a small part of the chart. If we zoom out a little bit, let's see, zoom out. There we go. You see a larger part of the chart, and it's just full of hundreds of different organic molecules and enzymes changing one into another. I mean, it looks like the Los Angeles City road map or freeway system or something. Anyway, so the cell is, as I said, the cell is full of thousands of different organic molecules, and there are thousands of different enzymes always converting one to another 24 hours a day. The word metabolism means all of those. The word metabolism means the sum of all the thousands of chemical reactions that occur inside an organism. Oh, I've gone over time, I guess. well, actually, I've got one more minute. Anyway, so all those chemical reactions going on are called the metabolism. And the people who study metabolism subdivide it into two parts, catabolic reactions and anabolic reactions. And in a nutshell, the catabolic reactions are the ones where the enzymes take larger molecules and break them apart into smaller bits. And anabolic reactions are the opposite. They're the building up reactions where the enzymes take smaller molecules and link them together to make larger molecules. All right, that's probably a good stopping point. So I will see you at 1 o'clock today, parking circle.